Everybody's online. We're here. We're good to go. And so, good morning, good afternoon. Welcome, everybody. Thank you so much for joining this webinar series. This is part of as a way of background, part of a webinar series. It's a collaboration between several of the Delaney collaboratories. It'd be RID HIV, DARE, CRISPR for Cure, and HOPE, among others. And then our friends at TAG, like Richard Jeffries and others who've assisted with this. And we've been doing this for what is it, 3 years now, Michael? Yeah, and it's we've had some really amazing programs, and today is going to be another one of those that we're really excited about. Loreen Willenberg is a person that probably needs no introduction to most of you on this call. She's known as a San Francisco patient, but for those of us who know and love her, we know her as an amazing activist. I first met Loreen, gosh, at least 15, if not longer, years ago in San Francisco, who just showed up out of the blue at a AIDS Treatment Activist Coalition. Nobody knew who she was, and she just kind of stepped up and said who she was, and talked about the amazing work she's done with the Zephyr Foundation
I don't know. The
and if
schedule
we could
doesn't.
ask people to please
There's
mute
no
if you're not
schedule.
speaking.
So I'm going after this.
Thank you. And we got to know and love her for all the amazing work that she's done as an advocate for elite controllers and for research in this area. So, without further ado, we're going to hear all about Loreen from Loreen, and then from two researchers, Michael Peluso at UCSF and Shuyu at Harvard. And so, we're really excited to have them all on board today. And without further ado, Loreen, we'll have you take it away and tell your story.
That's wonderful, Jeff. And you know what? I'm so glad you remembered when we first met, because I've had a hard time remembering myself. And you're right, I just kind of dropped into a meeting. But you know, what was interesting by that time, I think that I had, under the tutelage of Bruce Walker, as I would take those trips to, donate cells to their study. This is pre-Reagan Institute days. I think that they had received some funding from Bill Gates with the original cohort of HIV controllers. The more that he educated me about the uniqueness of that population around the world, I had to think, where's the best place to be to share this information, and also to learn more about treatment, HIV medications in particular. And I'd heard about ATAC. I learned about the meeting. And so I walked towards all of you activists to learn more about the medications, actually, even though by that point we knew that I did not require them clinically, right? And so that was very interesting because, I was a landscape designer. I was a botanist, running my own company in the foothills of the Sierra Nevada mountains where I lived at the time, and eventually became a landscape contractor to install those designs that I was doing. But I was kind of on a clean slate in terms of advocacy, and I realized that I was being pulled into a world that really captivated me, not only from a scientific perspective, because I've always, been a curious, lifelong learner, but how it was that HIV controllers, what they knew about us, 20 years ago, could be helpful to the community at large. And I figured, well, I might as well jump into the fray and meet some of these people that I had only really read about in Pause magazine. I had a subscription to Pause since the middle 90s.
Sorry.
Oh, that's okay. And God bless all of you. You kind of took me underneath your collective wings and taught me about treatments, the medications, and I started attending more and more of our meetings. We'd meet like in Pennsylvania, San Francisco.
Oh.
And it was very intriguing to me to sit in those meetings and listen to all of you speak in such educated ways about pharmacology. I didn't have a background in any of this. And here we are 20 years later, still endeavoring the work. And I'll always cherish how you all embraced me. And I knew that I had to walk towards you instead of away. And I've never regretted that. And that's why I'm smiling with each familiar name that I see coming up on this webinar. And you just all don't know how grateful I am to all of you for sharing your knowledge, just like the research scientists were on the scientific side of things. And
just keep yourself hydrated and rinsed.
Could everyone please make sure to take a moment right now, make sure you're muted. We've had like 4 different people come off mute by mistake, so please make sure you're muted. Go ahead, sorry.
Thanks, Michael. No problem, honey. So I think the most important thing I wanted to say was, as I found my footing in the advocacy and I started realizing that a nonprofit was a good idea to represent HIV controllers from around the world, I started building the Zephyr Foundation with the help of several friends back in 2006. We launched our website for the Zephyr Foundation in 2008. A good friend of mine, the webmaster of our site, also created a firewalled, highly encrypted forum for HIV controllers from around the world. I was getting emails from people. I was getting messages from people via different channels online. Folks who perhaps had been positive for 20 plus years, had never disclosed their status to even a family member. Every day was like a gift to me because I was actually in direct contact with the very individuals that Dr. Walker and researchers at the NIH were looking for to build their repositories of samples. And eventually we had 80 members sign up for that interactive forum. Why was that important? Not only could I help to refer them to studies in their individual countries, the more I learned about the study of HIV controllers around the world, eventually there would become, I believe, 20 different countries who had cohorts to study us. But it was that personal connection between people who had no other way to connect and discuss the phenomena of what it felt like to be a person who perhaps didn't require the medications or just needed someone to talk to about lived experience and same experience. And, I did all this from that blank slate. I think the most important thing was developing partnerships not only with researchers and the participants of their studies, but with other advocates to get the news out that we were not unicorns, because there was a lot of skepticism surrounding the HIV controller existence. And I remember when we were building our website, you could do a Google search and never get a hit on the term HIV controller, or this overlapping group that's very similar to controllers called long-term non-progressors, which are individuals who never progressed to AIDS after, I think the folks at NIH at that time were saying 7 years positive, without any progression toward AIDS. And so here I was, an individual with a company, but then I started to travel more, really captivated by this work and this connectivity. And I retired. I retired from my company to do this advocacy work. My family, my sisters thought I was a little bit crazy because I was quite successful and I loved my work. But there was something in my heart that told me, no, someone needs to kind of act as the poster child for this group because of the skepticism. I wasn't the only one that was exposed to skepticism, but I wanted individuals to know, look, if there's a group of folks out there that don't have any clinical signs, no symptoms of illness, then don't you think it's worthy of a really deep look at why that is true? And eventually, the news was getting out, although I do get surprised today, as recently as this morning, to hear Michael Luella say that at a recent conference, perhaps, or a meeting he was attending, that a vaccine expert had really no knowledge that myself, with the nickname the San Francisco patient, existed, or the Esperanza patient, which I think is a wonderful story. The town she lives in is named Hope. It's just extraordinary that there's two of we women. There might be a third that they're looking at. I believe the third woman, there was a recent paper published a couple of weeks ago out of Spain. They think they might have a third woman who might be an exceptional elite controller, or this upper tier that they kind of plug us into as having extraordinary control over HIV. So that's just a little background at my beginnings. I've loved the work. I'm on a separate health condition journey now. I am a cancer patient, but I'm doing very well. And she will perhaps address a very interesting conclusion that she and her colleagues back at the Reagan Institute have come to when it comes to cancer and my immune system. But I want people to understand that it's a group of very rare individuals. We are still very difficult to find. But I think with having helped somehow built the number of individuals who were participating in not only the Reagan Institute study of HIV controllers and other countries' studies of us, that I'm grateful that in some small way that I have been able to contribute to the knowledge that they have about our immune systems. A lot of folks are really curious too when I speak about how I felt when I was originally diagnosed with HIV in 1992. And obviously, it was very traumatic. It's not news that you ever expect to receive. But a very interesting point about my case and my very first HIV test. And of course, they didn't have the sophisticated testing methods that they do today. My original test actually came up as indeterminate. And I remember talking to the public health nurse who was, giving me the news about the results of my first HIV test. I said, indeterminate, what does that mean? She said, you're in between. You're either positive or negative. We need to redo the test. At that point in time, I had to wait 2 weeks to get the test results from the second test. And that test came back a definitive positive. So already back then, I was starting to learn some different names, some different terminologies. I always thought it was very interesting. I remember Dr. Walker said to me back in the early 2000s that it's too bad that the public health department up in the foothills didn't keep, didn't retain the sample, um, that might have been informative in the beginning. So I've always felt that that was very interesting. But the fact of the matter is, throughout the course of my, accessing medical care, with a doctor in Sacramento who tracked my labs like every 3 months for the first 5 years, it was he who actually told me, you seem to be part of a group that's very uncommon. You're having an atypical reaction to or response to HIV infection. And in 1995, my doctor told me, in the future, somebody's going to want to study you. I've just not heard of a study that, is being conducted to refer you to. And here comes an article, interestingly enough, by our friend and colleague Richard Jeffries for Pause Magazine. He had written an interview. He had done an interview with Bruce Walker. And that article is what alerted me to call my doctor in Sacramento and to contact Bruce Walker back east and say, I think we have an individual that you might need to study. And that's what started. This was 2004. This is what started my journey as a participant into clinical research studies. Dr. Walker's study being the very first. And Richard, I don't know if you remember that article. I used to have a copy of it, something about um, odd or something about the immune system. I'm sure you have it, hon. But it was in Pause magazine. That's how I found out that there was indeed someone studying folks like me. And that was the beginning of my walk. And I'm so proud to say that my last donation to that study before I actually retired as a clinical study participant, um, I almost lost my train of thought there. It's been 20 years. That's a lot of cells. It's a lot of blood draws. It's a lot of sigmoidoscopies to donate tissues from my gut. And, I've never really, until recently, I've never really felt the impact of what I've donated, but I've been happy to do it for the benefit of many. So I don't know how best to tell you that it's my hope that somehow, in all their dedication and brilliance, that researchers like Shu Yu, Bruce Walker, Mark Connors at the NIH, Michael Peloso, who's on this call with us today, will be able to define what mechanism, how did this happen? What did my immune system do that other people's immune systems need to do to keep them safe from developing AIDS? And it's my prayer, actually. I think about community every day, just like when I walk towards the treatment activists, that somehow they're going to define that action and share it with 40 million people in the world. That's what I live to hear. And that's why I know that my work on the earth isn't done, because wherever I can speak of this gift and this blessing and give all of you hope, give everyone hope that an answer will be discovered, and this uniqueness that myself and the Esperanza patient and other individuals called HIV controllers will be shared with individuals who won't need to take the toxic medications for the rest of their lives to survive.
Thank
So
you, Loreen.
I'm always curious about folks' questions. I'm happy to answer any that people have. But thank you. I'm just profoundly grateful for everything that you've all done. I respect and admire and love you all. And so I'll just wait for some questions, but let's listen to our researchers.
Exactly. Thank you. That's a great segue. Next up is Dr. Michael Peluso from University of California.
Michael, could you raise your hand, please, so I can find you?
Oh, thanks,
There we are.
Justin,
So we
for
can just
pushing me.
put you back in the spotlight.
I raised my physical hand and realized that that was not what you were asking.
Yes, no, thank you. Thank you.
Okay.
All right, so Jeff or Michael, you're gonna share the slides then?
Yes, I'll progress. You
Okay.
just say when.
Okay. Yeah, thanks for sharing that, Loreen. It's always great to hear your take on kind of the long journey that you've had, and it's quite inspiring, especially to some of the more junior people like me who are kind of at the beginning of our careers. And there's just so much important history. So thanks for letting me be a very small part of that. Yeah, so my name is Michael Peluso. I'm a HIV doctor and researcher at UC San Francisco, and have been here for about 8 years now. I came to UCSF with the goal of really understanding um, sort of how the HIV reservoir persists and why HIV is so difficult to control and cure. And as part of that focus, I have done a lot of work trying to understand controllers, especially these exceptional controllers like Loreen, and then also work on leading HIV cure focused immunotherapy trials to try to see whether we can, um, you know, move people's immune system from an immune system that is, not able to control HIV to an immune system that is able to control HIV. So I'm basically going to provide a sort of big picture overview of the current sort of thoughts about how a very small subset of people are able to control the virus without medications. This will put some context around what Loreen just described as her story, and then hopefully we'll set up what Ju will present next, which will be a little bit more detailed about the biology. So when we talk about when we're using these terms like elite control or natural control or spontaneous control, these are all sort of synonyms that are used to describe the same clinical phenomenon, which is people who are able to control the level of HIV in their body and in their blood without requiring HIV medications. And the first point to make is that this is really extremely rare. So there's not great statistics on this, but it's thought that fewer than 1%, and honestly, fewer than probably 0.5% of all people with HIV are able to control the virus without requiring HIV medicine. So what you're looking at in this schematic here um, is, time on the bottom on the x-axis and the amount of HIV in a person's blood on the y-axis. And the vast, vast, vast majority of people living with HIV are, what's shown in red here, that over time, once they acquire HIV, the amount of virus in their blood shoots up, and then it comes down to what we call a set point, which is a level that's typically between, maybe 10,000 copies and as high as like 1 million copies per milliliter of blood. And so that's the vast majority of people with HIV. When we're talking about elite controllers, these are people who, once they acquire HIV, it's thought that initially there's a little bit of virus that can be detected circulating in their bodies, although we often don't, capture that time period. And then this comes down probably over the first few months or first year, often before people are even diagnosed. And then they achieve a set point where the virus is really not detectable at all in their bloodstream. And there, there's a whole spectrum of people in between these kind of two extremes. For a long time, we identified people who were kind of viremic controllers who would achieve levels of virus in their blood that weren't, 10,000 or 1 million, but also weren't zero. There were, maybe 500 or 1,000, and those are sometimes called viremic controllers. So there's a whole spectrum of control. But what we're really interested in talking about today is what's shown in blue, these spontaneous controllers or elite controllers. And the reason that we, you know, really care about this is not just because it's interesting and a unique population. It's because we think we can learn a lot from these individuals in terms of what the body and what the immune system needs to do to achieve this level of control, and that can inform what we are trying to achieve in the clinical trials of the type that Steve Deeks and I lead, to try to get most people who can't control HIV without meds um, to that scenario where they are able to do that. And so we think that controllers, and especially exceptional controllers like Loreen, are are sort of a model for what we need to try to achieve in these studies. Next slide, please. Oh, can you go back? Thank you. Yeah, okay. There we go. So, one important point that Lorraine sort of alluded to is that it seems that women are much more likely than men to be able to achieve this sort of spontaneous HIV control. And as Lorraine mentioned, the two exceptional controllers that have been described so far, Lorraine, the SF patient, and the Esperanza patient, are both women. And that's really interesting, right? We know that there are important biological differences between women and men with HIV. Compared to men, women with HIV typically have overall a lower set point and a smaller HIV reservoir. This is especially true of premenopausal women. We think that there's a lot of interactions between hormones like estrogen and the ability to control HIV. And we know that women in general, not just women with HIV, but women in general, tend to have stronger immune responses than men overall, especially in the context of a new infection when they're first acquiring an infection. And I think, one of the best examples of that we've all lived through over the last 5 years is COVID, right? Where women who get COVID, when they get COVID, have a much more robust immune response in being able to control the virus in the first few weeks of getting a COVID infection than men. And that's probably why men, sorry, had tended at the beginning of the pandemic to get a lot sicker with COVID and to have a higher mortality rate from COVID. And so there are important characteristics of women that may make them more likely to achieve good control of HIV. Next slide, please. Is there one before this? I can't. Let me see. There should be one before this, but maybe it got lost.
Yeah, possibly. Go ahead.
Okay, yeah. So I'll just I have it on my screen, so I'll just describe it. So, the next question that sort of arises, this is not what is shown on this slide, is, a question about, what sort of biological factors are actually associated with HIV control. And there's been a ton of work over the last decades trying to understand exactly what is going on biologically in elite controllers. And the upshot of all of that is there are a few things that we really understand quite well, which is that elite controllers tend to have really, really potent and robust and sustained CD8 T cell responses against specific regions of HIV that are particularly vulnerable to the immune system. So CD8 T cells, right? We think about CD4 T cells in HIV as helper T cells, and those are the cells that are really knocked down by HIV. That's where HIV lives, and people lose their CD4 T cells over time. Um, but it's the CD8 T cell response, the killer T cells, um, that seem to be really sort of special in elite controllers. And there's a lot of work trying to understand why that is and how that can be sustained. The other thing that seems to be um, more unique about elite controllers is that many of them have special genetic characteristics, and so that's something that our genetics are something that we all have since birth. And there are certain types of genes that are thought to be protective in HIV. These are genes like HLA B57 or B27. So those people who are able to control HIV without meds are much more likely to have those kind of specific genetic markers. And that is likely to influence their immune response. Um, that can be a double-edged sword. Some of those genetic markers that we see more commonly in HIV controllers are actually associated with autoimmunity, conditions that can cause the immune system to kind of go haywire. And so it's not as straightforward as certain genetics are good and that makes people more likely to control, but it's part of sort of the bigger picture here. And then there are lots of other factors that we really don't understand as well that we're trying to kind of over the years by studying lots of these people get a better sense of. So now what's actually shown on the screen is very important. So what happens to most elite controllers over time? So elite control is not it's an ideal state to be in if you're living with HIV, but it's not a perfect state. And so what I mean by that is over the last 20 years, we've learned a lot from studying elite controllers. We've learned that many of them have ongoing HIV replication, and so even though uh, the amount of HIV in their bodies and the level of replication might be lower than the average person with HIV. It's not in every case no replication. And we know that HIV replication can have consequences. And one of the main consequences that we really care about clinically is inflammation. And so we know that um, HIV, many HIV controllers who are not on meds over time have higher levels of inflammation in their blood and in their bodies and in their tissues. Um, and that inflammation can have consequences. We also know that many controllers lose control over time. And so they may have control for 2 years or 5 years or 7 years or 10 years. But what makes people like Loreen exceptional is that they actually are able to maintain this control for decades. So among that 1% of people with HIV who are able to control without meds, there's probably an even smaller percentage of that 1%, maybe 10%, 5%, 1% who are able to kind of sustain control without, we think these consequences. And then the last consequence that we really care about, are those types of consequences of losing your CD4 T cells over time, of inflammation for years or longer, which is really things like cardiovascular disease. So, what we're talking about there, and Peter Hunt and Priscilla Hsu at UCSF have both done a lot of work on this, is higher risks of heart attacks or strokes, or other inflammatory consequences. And so anyway, the result of all of that is that even having a lower undetectable viral load without meds in many cases is not the same as not having HIV at all. And the result of all of that, especially as HIV meds have become so much less toxic and more tolerable and easier to take over the last 30 years. Especially, with many people now being able to be on one pill once a day, is that the standard of care is to encourage everyone with HIV, even people who are controllers, to go on HIV meds because we know that can often reduce the level of inflammation, and have benefits over the long term. The other point that I want to make is that it's actually a lot harder now to identify controllers, because the standard of care, since 2010 to 2015, depending on where in the world a person is to treat everybody as soon as possible. And so,
Right.
We've been able to identify controllers because the standard of care in the 90s and the early 2000s was not that, and people would be off of meds until they progressed. And so now that people who immediately upon diagnosis are getting started on meds, we don't have that opportunity to kind of observe people over time and see them achieving control and sustaining control. So this is a lot harder to study now, and we're very fortunate to have these, long-term survivors, long-term non-progressors, controllers, who were identified in the 90s and 2000s, to really understand this. Next slide, please. Okay, so as Loreen mentioned, some elite controllers are exceptional and they don't have that progression that I just mentioned on the prior slide. And that's really why we're here today and identifying these cases. Loreen was the first of these cases really to be deeply characterized. Um, is really important in us understanding kind of the full spectrum of HIV. Um, there's not a textbook definition for this yet because we're just starting to understand it, but generally it's thought that exceptional controllers have very low reservoirs, and Ju will talk about this, have, stable CD4 counts, don't progress clinically, as I mentioned, have very low levels of anti-HIV antibodies, and in some cases, people, lose their HIV antibodies. That's okay. These antibodies don't protect against HIV. They're a diagnostic test, really. And it's quite interesting that likely because of the very, very low amount of HIV, in some of these controllers' bodies, their antibody response just drops off over time. And then we also think that these individuals have overall low levels of T cell activation, probably because their immune system is not being stimulated because their levels of HIV are so low. Next slide, please. So in the last few minutes, I'll just comment on what is being done to really understand controllers now. So, and there are 5 points I'm going to make. So the first point I'm going to make is that we and others are doing studies to really identify controllers, not only that look different from most people with HIV who are on meds, but who actually look closer to people without HIV, right? And so what you're looking at on this slide is some data that we presented at IAS last year, where we studied, and this was in collaboration with Ju and Matthias at the Raygon. We studied people with HIV who were on HIV meds, shown in green. we studied elite controllers shown in blue, and we studied people without HIV who were at risk for HIV, shown in red. And each little rectangle on this represents an individual person. So there are like 150 little colored rectangles on this, right? And what I want you to look at here is how the colors group together. So, it seems that they're kind of randomly distributed throughout here. But if you look really carefully in this red box in the middle, you can see that a lot of the people without HIV, the red, are all grouped together, right? So they all look similar in terms of, we measured like 1,000 different things in their blood, different proteins for inflammation, immune activation, etc. So you can see that many of the people without HIV group together, but interspersed in that group are a few blue rectangles. So those are elite controllers who have HIV, but in terms of what we can measure in their blood, are indistinguishable from people without HIV. So that is suggesting that a subset of these controllers immunologically look no different from people with HIV. That's really, really interesting to us. And so we're trying to understand that. You'll also notice that there's really no green interspersed in there. So most people with HIV who are on HIV meds are not able to achieve a state where they totally resemble people with HIV. Okay, so that's one point. So next slide, please. Oh, okay, that's the slide I was trying to talk about before.
Oops,
so that
sorry.
got mixed up. That's fine. That makes me feel better. At least we know where it went. Okay, the second thing that we're trying to do to understand controllers is to look at controllers who are able to maintain control versus who lose control. And a lot of this work has been done by Bruce Walker, who Loreen mentioned. He's a senior scientist at the Reagan Institute, and his colleague David Collins, who's a more junior scientist like me. And what they have found in a really beautiful study that was published in the journal Immunity last year, they studied about 30 or 40 controllers, some of whom were able to maintain control for years, and others who we witnessed kind of lose control over time. And what they really found was that it was the CD8 T cell responses that were either maintained or lost over time. That's these CTL responses that distinguished those who had durable control versus those who lost control. So a lot of interest in understanding whether we can predict the subsequent loss of control. Okay, next slide. The third thing that we're trying to do is see if we can understand how controllers initially achieve control. And so, as I mentioned, it's really hard in this day and age to study people who are achieving control because almost everybody gets started on ART on HIV meds immediately. But we at UCSF realized a few years ago that we were in a very unique position because we had a large cohort of controllers who had gone on HIV meds for all of the reasons I mentioned a few slides ago, increased inflammation, consequences of having any amount of virus. And a lot of those controllers were willing to come off of their HIV meds to see what would happen. And so with support from the Gates Foundation and a study that was designed by Lily Cohn and led by Becky Ho, we did this very, very highly controlled treatment interruption where we paused people's HIV meds. So what we did was we worked with people who were on HIV meds, some of whom were, regular people with HIV who were just on meds, and some of whom we knew were controllers before they started meds. And we measured them really carefully, took lots of blood samples, tissue biopsies, et cetera, while they were still on meds. And then in an extremely controlled and coordinated manner with a lot of help and advice from our community advisory board and Corinne Dubé and our social science team, we interrupted their meds and watched what happened. And for most people, when their virus came back, as soon as we got any sense that there was virus there, we resumed meds. But for some people who we knew were previously controllers, we watched them to see whether they would control again before restarting meds. Next slide. When we, is there one before that? I lost another one. Okay, this one, yes. Go forward again. Go back. Sorry, this is all scrambled. Okay, yeah. So what we saw in red were people who were non-controllers, who their virus came back really quickly over the first, 10 to 14 days. But then what we found in blue in the prior controllers is that their virus came back, but then they were actually able to re-achieve control. Some of them were controllers at levels of 500 or 1,000. Others were controllers at levels that were undetectable. So this was fascinating. We watched them re-achieve control. And then now we're doing a really deep dive to understand biologically how they did that. Okay, next slide. I'll just be very brief on this point, which is that actually, when their virus initially came back, when we compare people who were prior non-controllers to prior controllers, we actually see that the speed with which their virus comes back when they pause their meds is very, very different. You can see that the slopes of this red line versus this blue line are very, very different. So what that signals to us as researchers is that actually there's probably immune system changes that are happening really in the very first days after the virus and the immune system interact with one another that determine whether someone is destined to go on and be able to control. And we're doing a lot of work now to understand what's going on, biologically. Okay, next slide. It should be the one that's number 4. Okay, so the fourth thing that we're doing, and I just have one more after this, is really trying to characterize this growing cohort of exceptional controllers. Like Loreen, this is Loreen giving a talk at an event sponsored by AmFAR a couple years ago, which was really wonderful. The description in Ju's beautiful paper from 2020, the Esperanza patient that Loreen mentioned, and Ju will talk in her segment in a lot more detail about the characterization of this group, but understanding what's going on biologically in this very special subset of controllers, exceptional controllers like Loreen is so important. And my last couple of slides are about trying to, basically achieve this, thinking about controllers as a model for HIV cure and trying to move people's immune system in clinical trials toward a controller phenotype. So I won't spend a lot of time going into this, but I just want to provide a broad overview. This was a study that was funded by AmFAR and is going to be published on World AIDS Day next month, where we did a series of immunotherapy interventions. This was taking people who were not HIV controllers, just typical people living with HIV on meds. We did a variety of different immune system targeting interventions to try to alter their immune responses. And then we paused their HIV meds and observed what happened. Next slide, please. This is similar to what I showed before. So, I showed you before the red and the blue from that other treatment interruption study. And I said that the blue people were controllers. What we saw in this immunotherapy trial is that we were actually able to alter people's immune systems so that the green lines here overlap with the blue lines, right? This suggests that we're able to alter people's immune responses with this very intense series of interventions to get them, when their virus comes back, to kind of look like what the controllers did. And so understanding this overlap and what might be causing this is really, really important. And this is how we're thinking about spontaneous controllers, natural controllers, elite controllers people like Loreen as a model for what we're trying to achieve in HIV cure studies for kind of everybody else with HIV. Okay, I think that, and then, yeah, okay. And then the last thing I'll say is that, and this also sets up Zhu, is that we have, in the context of these cure studies, found people that we think look quite similar to Loreen. This is a participant in our study funded by AMFAR who was treated very early for HIV infection, basically immediately started ART, HIV meds within a few days of diagnosis. Participated in that immunotherapy trial that I just showed you, had no rebound for over a year and a half, eventually resumed HIV meds, not because of any specific reason related to HIV, but just, it's a lot of monitoring to be off of meds for such a long time. But then again, with AMFAR funding, in a program led by Zhu that we're participating in, is now in the midst of a second treatment interruption, and again has not rebounded for nearly 6 months now. And so we're wondering whether this individual is sort of another example of a very exciting spontaneous or treatment-induced cure. And Zhu will talk more about that. So I hope that that provides background for what Zhu will talk about in more detail, and I will turn it over to her now. Thank you.
Yeah, thank you, Michael. Many thanks to Jeff and Linda, as well as all the CAP leaderships for this invitation. So Loreen and I actually supposed to host a session together in the AIDS 2020 in San Francisco, but right around the time we actually first reported her as the first person to have achieved a natural cure of HIV infection. But unfortunately, as you know, due to the pandemic, that session was canceled, which means we did not get the chance to meet each other in person until now. So today is the first session we both are in together, although it's still not in person. And it is a real privilege to be part of this discussion with Loreen, with Michael, and with everyone, with the community audience here as well. So next slide, please. So today I'm going to walk you through one of the most remarkable scientific stories in the field of HIV cure research. And then through the story, I would also like to share how we define what we call a virological cure of HIV infection and what makes someone an exceptional ED controllers in our opinion. Next, please. Yeah. So let's start by discussing how HIV behaves in the body and why finding a cure has been so challenging. This is basically normal human cells. So DNA acts like a blueprint. It copied into RNA, and then RNA then used to make proteins. And this is the usual flow of information in our human cells, allowing our cells to function and reproduce. Next, please. Yeah. However, HIV is a retrovirus, which means it does things a little bit in the reverse direction. So HIV carries RNA as its own genetic material, and then once it's entered a human cell, it converts that RNA back into the DNA. So that's important because most of the virus actually never becomes part of our own human genome, but HIV does. It takes its RNA and turns it into DNA, and then next slide, please. It insert that. No, sorry. Yeah. Then it's insert that DNA into a human immune cells, which is usually the CD4 T cells. So once this happened, HIV is no longer just a virus floating around in our bloodstream. It becomes part of our infected cells' genetic code. So we call that integrated form of the HIV is a HIV provirus. So at this point, Once HIV becomes a provirus inside the cell, it can stay there for the entire life of the cell and even pass into its daughter cells when the cell divides. So that's why HIV infection can persist for a lifelong. Next. With the application of the next generation deep sequencing technology, we now have a much better understanding of the different types of proviruses in the whole cell genomes. So the good news is not all proviruses are the same. Most of the HIV proviruses we find in people are actually broken or defective. So you can think of them as the fossils, viral fossils. So they are basically reminding us that infection has happened, but they cannot produce new viruses anymore. Next, please. But only a small fraction of proviruses that integrated into the host genome are actually intact, meaning they have all the genes HIV needs to wake up, to make new virus, and to restart infection. Even though these intact proviruses are very rare during treatment or in many of these natural controllers, there may be only one in 100,000 or one in a million blood cells, but they represent the long-term risk. So when we talk about curing HIV, we're really talking about eliminating or permanently silencing these intact replication competent proviruses. Next. Good. So we know that ART can completely suppress viral replication by blocking quite a few different steps of the viral life cycle, but it actually cannot remove the integrated HIV proviruses in the reservoir cells. Next, please. So when we talk about a cure, for most of the other infectious disease like bacterial infection or flu, we usually mean that very simple, all infection is gone. All the germs, all the infected cells, all the traces of the pathogens are eliminated. But for HIV, because it's a retrovirus that inserts itself into the host genome, we need to make a quite important distinction between different types of cure. So the first type of cure, we call it a virological cure. And that could mean the complete elimination of all HIV infected cells so that no trace of HIV, even those defective pieces, can be found, which will be very, very difficult. Next, please. So to a lesser degree, a virological cure can also mean that all intact replication competent HIV reservoir cells are eliminated. So no intact viruses anywhere in the body, but some defective proviruses may still exist. Next, please. But there's also a second type of a cure. We call it a functional cure or durable remission of HIV infection. So here, some intact proviruses still can be detected and exist, but the immune system or other mechanisms keep the virus permanently suppressed without the need of therapy. So with, I'm not going to go into the details, but with the analysis we have done before, we now know that these intact proviruses are often integrated into those so-called gene desert area in the host genome, where they are much less likely to be reactivated. So this may explain why these intact cells can be silenced for long term in the absence of the therapy. But again, we're not going to go into this today. So today we're going to focus on the type 1 virological cure. Next, yeah. So we all know very well that so far there's a growing list, but still very small number of people who are considered to have achieved the type 1 cure of HIV infection after stem cell transplantation, usually really performed for cancer treatment. And these cures involve donors who have a rare genetic mutation, the CCR5 delta 32, which makes the cells most resistant to HIV infection. Then all these cure cases showed no viral rebound after transplantation and after treatment interruption of the ART. But now what I want to emphasize the laboratory evidence that we used to confirm these cures. Next, please. So instead of relying solely on the standard viral load blood test, quite a different group of investigators have used the multiple extremely sensitive assays, testing millions to billions of cells from blood and tissues. So what we see here is several type of laboratory method. So you can see here there's a qPCR means quantitative PCR, and ddPCR means digital droplet PCR. So these are the assays to detect the DNA fragments encoding different parts of the virus in the human genome. However, these methods cannot tell whether these viral DNA are defective or still intact. And the viral outgrowth assay, on the other hand, they actually is specifically designed to detect the replication competent proviruses. And these are the viruses that are actually capable to produce infectious viral particle. For example, this is the oldest data summarized for the Berlin patient. So no replication competent HIV was detected even after 1.4 billion CD4 T cells were tested using the viral outgrowth assay. But if you see it, there's also one qPCR test showing a trace of HIV LTR region, which might detect some defective proviruses. So overall, you can see an extremely extensive and rigorous evaluation was performed, and it confirmed that the Berlin patient was the first person in history cured of HIV. So this level of thorough investigation really sets the gold standard for defining a virological cure, in my opinion, at least. So next. So for this is the summary of all the laboratory analysis done for the Linden patient. Again, multiple assays were performed, but this time the number of cells are much less actually analyzed in these. You can see here no replication competent viruses were detected in 24 million resting CD4 cells, and a little bit similar to the Berlin patient case, a few of these vials actually showed weak signals of viral DNA application in the ddPCR assay. But these were probably defective proviruses, but these assays will not be able to tell. Go ahead, next one, please. Yeah. So, but we also know, as Loreen mentioned, she had donated so many different tissue samples. HIV can persist in tissues such as gut, lymph node, brain, and reproductive organs. So for the cure cases, investigators also went even further. They analyzed gut samples and lymph node samples from these patients. Once again, most of the tests were negative, with a few exceptions. Some rectal biopsy samples from the Berlin patient and one lymph node samples from the Linden patient showed very low levels of HIV DNA signals again. However, again, this is done by ddPCR and most likely come from the defective viral fragment present at an extremely low level. Go ahead, next one. So what we just reviewed is the treatment-based cure achieved through transplantation. Now, so I'm going to really emphasize these remarkable cases of natural cure. So they did not have any treatment, not transplant, not even taking any antiretroviral therapy. So we call these individuals as exceptional elite controllers because they really may represent a natural cure of HIV infection. So the first person we all know is Loreen, who at the time of the publication we refer to as the San Francisco patient. Just very brief revisit of the clinical history Loreen already mentioned. She was diagnosed with HIV in 1992, have never taken ART. And at the time of our analysis, she has, for more than 24 years, had no detectable viremia except one exceptions in, I think it's January 2000. For her case, we used even more sensitive assay to look for HIV DNA. So that's the full genome sequencing assay using the next generation sequencing technology. So you can see here on the table, we analyzed the 1.5 billion blood cells and found no intact HIV DNA sequences. We also analyzed the 4 million gut biopsy cells using the same deep sequencing assay. Again, no intact proviruses were found. And we also performed the digital droplet PCR on 14 million resting CD4 cells. No intact HIV DNA were detected. And with the traditional viral outgrowth assay, we analyzed over 340 million resting CD4 cells and 41 million total CD4 cells, again, showed no replication confident virus. So this really represents the same level, if not even more extensive levels of search for intact or replication competent HIV as what has been done for the Berlin patient. And on this slide, actually, interestingly, using the next generation sequencing assay, we now actually can detect the defective provirus very well. So we detect a total of 19 HIV DNA sequences among the 1.5 billion cells analyzed from Loreen, and most of them have large deletions, and 4 of them actually is full length, but they are heavily hypermutated. So as I mentioned earlier, these defective proviruses are like viral fossils. So it's evidence of the past infection, but they would not be able to produce any new viruses anymore. Next. So the second case is the Esperanza patient. She was diagnosed with HIV in 2013. Again, never took ART, and then her viral load has always been undetectable. So using our new highly sensitive full genome sequencing assay, we tested about 1.2 billion blood cells and more than 500 million placenta cells, and mostly also blood in there. And again, no intact proviruses were found. And the traditional viral outgrowth assay was also performed on 150 million resting CD4 cells, and no replication competent viruses were detected. Next. Again, we did detect 7 defective HIV DNA sequences in the 1.19 billion blood cells from the Esperanza patient samples, consistent with her known history of HIV infection. Next. So now we have not 1, but 2 possible examples of natural HIV cure cases after extensive laboratory evaluation. And both cases reached the gold standard, if not more, set by the Berlin patient in terms of confirmation. However, even with these extensive data, we have to recognize that we did not analyze every single reservoir cells in the body. So as scientists, we can never be 100% definitive, but these are as close as we can get. Next. So I also want to highlight just how extensive our analysis must be to define someone as an exceptional ED controller or possible natural cure or possible virological cure cases. So if you look in details of our publication in 2020, where we reported Loreen case as the EC2, we actually also have an EC1 in that paper. And that's highlighted by this red arrow. And then the figures on the right is basically when we summarize all the ED controller data we generated after that initial publication up to 73 ED controller. You see both Loreen's case and Esperanza patient case. Again, this red arrow, we have this one participant that's the same as the EC1. So this is a male participant. We almost want to call him the Texas patient already. we analyzed 960 million blood cells and found no intact proviruses. But then when we analyzed the next 50 million cells, we detected one intact proviruses. So in this EC1 person, we had one intact in 1.01 billion blood cells. Therefore, we cannot classify this person as a possible natural cure because of the existence of the intact proviruses after massive analysis of the large number of cells. So this example really underscores that to define a possible virological cure, we need the absence of intact or replication competent viruses in over 1 billion cells analyzed by multiple highly sensitive assays in the laboratory. So that's the definition our group used to identify exceptional ED controllers. Next, please. Okay, so this is the case that Michael mentioned in his talk, the SCOPE participant 2523. So clinical data that Michael already mentioned, diagnosed with HIV also in 2013, but very early ART initiation, 12 years of recorded undetectable viremia, and had a post-treatment control for 18 months, and then now on a second ATI for 5 months or more. Here we have to say it's not a simply post-treatment control, but it's actually post-treatment elite control, which means no undetectable viremia in any time point after ART is interrupted. So we, again, in the process of doing very extensive laboratory evaluation, so far we have analyzed over 500 million blood cells using the highly sensitive full genome sequencing assay. We found 46 defective viruses, but no intact. And then we also performed traditional viral outgrowth assay on 143.5 million CD4 cells, which actually isolated from over 800 million blood cells. And again, no replication competent viruses were found. But the analysis has not reached the gold standard set by the Berlin patient, Loreen, and Esperanza patient, but it's on the right trajectory, but we're still very cautious before we call this person, who's a male, as the first person achieved a virological cure after treatment interruption. So next. So now I'm going to go back to really highlight Loreen's subsequent journey to become the first natural cure patient. So for Loreen, the HIV RNA has remained undetectable without any antiretroviral treatment for the past 6 years since our last sampling in 2019. So I remembered, So I remembered vividly that Loreen called me herself in the spring of 2023 to share the news of her lung cancer diagnosis. I was truly attached that she insists in telling me personally, but as you know, Loreen, her positive spirit has always been inspiring. And looking back now, she was absolutely right. So her immune system once again rose to the challenge. So I'm very happy to follow through all these journeys with her in the past few years. Next. So since her diagnosis, she has undergone multiple rounds of chemotherapy. We all know that chemotherapy are highly immunosuppressive. In addition, she also received anti-PD-1 antibody treatment. The commercial name is called Keytruda, which blocked PD-1 is an immune checkpoint molecule that we will discuss a little bit in details later. So this really created a rare and important opportunity. The question we had is, would HIV come back under these chemotherapy stress and PD-1 blockade, or would a cure hold? So in the standard clinical practice, for people living with HIV who are diagnosed with cancer, ART is usually started immediately if you have not been on one. But Loreen was able to convince her care teams that she did not need ART. but remarkably, her plasma viral RNA indeed remained undetectable throughout the treatment process. So at this point, I would like to replay Loreen's message to us in IAS 2025 conference. I hope you'll enjoy it one more time with me.
I am humbled by this remarkable news, and I assure you I'm doing very well. Please remember, miracles happen, and there will be a cure for both cancer and HIV.
Thank you. Thank you, Loreen. Thank you, Jeff, for playing. Okay, next. So now I'm going to tell you a little bit more about PD-1. So we know that CD8 T cells are the main immune cells that can recognize and kill both tumor cells and HIV-infected cells. So when they are healthy and functioning at full strength, they can produce anti-tumor and antiviral molecules. They can proliferate to make more of themselves, and they can directly eliminate or kill the tumor cells or infected cells. Next. But once these CD8 T cells has been working too hard against tumor or against infected cells, they become exhausted. So one of the hallmark of this exhaustion is the upregulation of PD-1 on their surface. So PD-1 basically act as a brake. They prevent excessive activation and cell death. However, while this brake helps the cell survive, it also causes gradual loss of their ability to clear tumor or infected cells. Next. So clinically, this is where the checkpoint therapy comes in. For example, treatment with anti-PD-1 antibodies blocks the PD-1 signaling on the CD8 T cells, then can effectively release this brake. They will allow this exhausted T cells to recover their function, to proliferate again, and to become active and kill tumor cells. So this strategy has transformed the cancer treatment and has been also being tested in HIV cure trials. Next. But the complication during HIV infection is that HIV-infected CD4 T cells themselves also show signs of activation and exhaustion. Next. Which leads to the upregulation of the PD-1 on these CD4 infected cell surface. Next. It has also been shown during ART, PD-1 signaling in the reservoir cells can actually reduce HIV gene expression and helping the infected cells remain in latent, non-productive state. And the effects actually help stabilizing HIV latency. Next. Therefore, when anti-PD-1 antibody is given, they may reverse this latency and reactivating proviruses that were previously silent. Next. Here is a list of 5 clinical trials that gave anti-PD-1 antibodies to people living with HIV, but all of them are on ART. So in the first 2 trials, Keytruda is the same drug Loreen received, was used. And even though all the participants were on ART, increases in plasma HIV RNA were consistently observed. Next. So now with all this information in Loreen's case, despite experiencing extensive immunosuppressive chemotherapy and anti-PD-1 therapy, which have the latency reversing effect, she has continued to maintain undetectable HIV viremia without the need of ART. Therefore, I believe this really represents the strongest evidence yet that Loreen is truly cured of HIV on her own. I hope you agree with me now that there's no longer a need of scientific debate whether a natural cure of HIV is possible. I think the question for all of us is how can we make HIV cure safe, scalable, and accessible to everyone who need it. So that's all I have to share, and I'm happy to answer any questions with Michael and Lorraine together.
Terrific. Thank you so much,
Awesome.
all of you, for your excellent presentations. It was amazing to hear Loreen's story in her own words and then kind of the scientific explanation of what that all means. You did that beautifully. So we'll go ahead and start questions. Technically, this call stops at 30 past the hour, but if people have time and want to stay on, you're welcome to do so. So we'll take it from there. And hopefully, Michael has been kind of vetting the questions because
Yeah, there's a lot of questions. I was going to invite those people who are still on, if they had a question that they wanted to feature, just raise their hand and let us know that way. Because if not, I'm just going to be picking in random questions. And so if you want your question to be featured, just kind of raise your hand so you can ask it yourself. But let me see. So, question on the non-intact proviruses, correlate of natural control, question mark. What are the mechanisms leading to defective proviruses and how do we encourage virus that path?
If I understand the question correctly, it means the question is about the defective proviruses,
Yeah.
whether they are still.
It's me, it's Patricia. I asked that question. Sorry, I was like trying to follow along and write. Yeah, I was really interested about your slide. You were showing all the folks that have those defective proviruses, and I was wondering if you can explain how that happens, because it seems that's something that all the folks that are able to spontaneously, control and cure have that. Is it a result of the action of the pressure of the immune system when the virus integrates? Is it linked to the specific place in the genome where it integrates? And if that is something that says, oh, they're defective, therefore they cannot reactivate, how do we encourage the virus down that way rather than being in a place where the virus is going to be all nice and comfy and can be complete and continues to, multiply?
No, yeah, actually, this is a kind of part of the retroviral biology. So retrovirus, they are smart. They carry RNA, as I mentioned, carry RNA, make themselves DNA, and insert them into the host genome. But fortunately, they are not very effective process. So that process actually a lot of error prone. So their reverse transcriptase can introduce a lot more error than our human polymerase, the one we reproduce our own cells. So that's kind of the defects of the retrovirus. During that process, they generate a lot of mistakes. They have a lot of deletions. They have a lot of hypermutation also happening during that time. So they still made that process to get into the host genome, but they are not always effective. So this is not only in controllers, in ED controllers, in people on ART, also majority of the HIV DNA that inserted in human cells are defective. Only few are intact. The problem here is even 99.9% of the proviruses in the genome are defective. As long as the few still left, sometimes stop ART, they can those intact proviruses enough to cause more rebound again. So that's the problem of the HIV cure we're facing.
Gus, you have your hand up.
Yes, hello, greetings from grim, grey, rainy London. I'm sorry, I missed the first part because of time zone confusion, so I'm afraid I missed Loreen talking, which is a great shame because I would love to have heard you. You are amazing and exceptional, but excitingly becoming less so. There's a couple of really interesting things. And by the way, I should also say that hello again, Sue, because interviewed her recently. I just come back from well, not just, a couple of weeks ago, came back from the European AIDS conference. Really interesting couple of presentations there. We now have Berlin patient number 2, who you may know about, who is heterozygous for CCR5, meaning that whatever's going on with him, I think it's a him, it's not necessarily to do with the fact that his cells can't be infected with HIV. They can. And he's the third one like that after the New York patient and the Geneva patient. And they found some really interesting stuff. Now, tell me if I've got this right, Sue. He had some sort of unusual natural killer cell reaction, which in turn set off um, an antibody reaction to HIV that's even stronger than they found in some exceptional elite controllers. So that's number one. And number two, I met a guy called Johan Mare, who is the guy who has stayed undetectable for 4 years after receiving a single shot of two BNABs in the Rio study, because he wanted to talk about his experience. So we're starting to see this trickle of really interesting cases of people who, for one reason or another, something's going on, which is helping to suppress their virus, and we're starting to find out quite a bit more about what's actually happening with them. I don't know if you've got a comment about that, or either of you, really.
Yeah. No, what you mentioned is really important. I think that's the beauty of getting that cohort getting bigger and bigger. So every case at this point, I don't think we know how they controlled and how they become so exceptional. But on the other hand, also we don't know which immune arm, whether it's unique to each individual, is a common among all of them that we actually can identify. I think that's really the next step. Now we have a cohort. I would say we have a really amazing the growing list of either a natural controller or the post-transplant controllers, cure cases. We actually can now go back to their samples to understand what's the common characteristics in the immune arms that we can learn from.
Mm. The exciting thing about Rio, I should add, is not just that there was this one exception, because there's always an n equal 1 in these studies. It's that there were lots of them in terms of people who actually stayed quite a long time off therapy. He's by far the longest, but there was a lot, lots of delayed rebound in that study.
I'd like to bring up the question that Jason just wrote. Can any of the scientists speculate on their most favored hypothesis for the mechanisms of protection? Exceptional CD8 T cell response?
Maybe I'll go first because I'm sure Zhu's answer will be more sophisticated than the one I can provide. But yeah, I mean, I think that it's likely um, the initial immune response to the virus at the time in which the virus is acquired that probably has a big impact on sort of the long-term trajectory, and then, maybe sets people up to then kind of mop up whatever remains over a subsequent, like, longer period of time. Um, but that is just my speculation. I'm not sure if you totally disagrees.
No, I don't disagree. But I think I think we're getting closer to mapping out what's going on, but I think we're still not fully understood different phase of the infection and what immune armors are playing a role. My sense is for ED controllers and exceptional ED controllers, because they're never on ART, I think a major arm of control might be CD8 T cells, because they can see the infected cells that have the HLA presenting epitopes to the T cell responses. But once ART starts, many of the cells actually will not be able to, the infected cell will not present epitope to CD8 anymore. So at that point, I believe this other immune arms, the antibodies or NK cells, would probably play a major role in that sense. But once treatment stops, during the ATI phase, whether we need a collaboration between NK cell and T cells, ANAPs, and to all contribute to that post-treatment control, or which arms play in the major role, I don't think we have a very clear answer yet. But different scientists, different groups are looking at each of these aspects currently extensively, with the cohort getting bigger for us to rely on to find the answer. I'm actually pretty optimistic for the next few years. We're getting closer to the truth, I would say.
Interesting. So there is a question. I'm trying to find it. I can't where to go. Here we go. Uh, They wanted to hear your thoughts, Loreen, on the terminology and how you would prefer to be referred to exceptional control or elite exceptional control. What do you prefer?
Oh my. We're going to go back years here. I remember hearing Bruce Walker use the term elite controller 20 years ago. We were in Toronto at a scientific conference, and I just told him, I said, that is such an off-putting term. You know, I think I understand why on your side of things that you need to use a term that separates some of us from, as Michael Peloso mentioned, those individuals called viremic controllers that might have a viral load that floats up to 2,000 copies per ml and then, stays there, never goes beyond it. But just from the street sense, the term elite, I mean, I really didn't like it. And so fast forward, now we have and you and I, by the way, that reminds me, several years ago, you asked me that question. We were trying to come up with different terminologies. And I told you, I remember sharing a list with Richard Jeffries over the Zephyr Foundation times. I think I had 43 different terms as of 2015 that, researchers around the world were using to label us. And I think we kind of talked about, you and I, HIV neutralizers, but then we couldn't use that because it described a completely different, segment of the population, and it gets all really confusing. Now we have exceptional elite control, and again, I have that understanding that sets, it's just a term that sets apart, a certain number of individuals like Esperanza patient and myself. I get it. I think Dr. McGillis from the NIH in a paper in the journal Blood, I think it was 2008 or 2010 maybe, where he talked about extraordinary control. I mean, I think that what it's done, these different terminologies, as we have, as activists and advocates and researchers alike, gotten to know each other better, it's wonderful that we're actually able to have these conversations with them directly. I know I have had with Shu Yu and Steve Deeks, with Bruce Walker too, that some of these terminologies to the layperson, those people who aren't like, science nerds like we've become, whatever, to be careful and mindful, and they are, because we don't want to alienate folks from community, right?
Right.
So I don't know. It would be kind of an interesting thing to do, is like have a survey, get some ideas from community forums, what would you think is appropriate, because from my standpoint, none of us do it intentionally, to exclude individuals. So, but I think it opens up a discourse. I think it's a very positive thing to have communication and discussion about what terminologies are people comfortable with. You know, and several folks on this call know that for years I resisted being called cured. Right? I always maintained cured from what? I've never had an illness, related to HIV infection, that kind of thing. But, folks, we're people together who are thoughtful and caring, and we don't want to exclude anyone from the conversation. We want to create environments of inclusion. That's what I try to do in these presentations. Compassion helps, kindness helps, understanding, tolerance helps, especially these days, where uncertainty seems to be the rule. But we can do this. We can come up with comfortable phrasing, I believe, as a community together. And that would be some of my thoughts. I hope that was helpful.
Yeah, I think it was helpful to just give a full reaction from one person who has one of these labels that might be offensive to other people, and to hear from you really helps set a record, of what people prefer or how to prefer. And it also I really appreciate that you kind of acknowledge other people's discomfort with it, and you're willing.
Absolutely. It's okay to be there,
Yes, definitely.
But some of the terminologies can be counter to what I try to represent, which is a story of hope. And I know that the Esperanza patient would probably say the same thing, as would any of the individuals on the list, like, our dear departed friend Timothy Brown, or Adam, the London patient, individual, gentleman I have yet the pleasure to meet. But we have to remember that we're all in it together, and we have a common goal, which is to witness, a cure. Yes.
Definitely. Is Mark Franca still on? Is he still there? I thought I saw his name still in our midst. Maybe not. Okay. Well, I don't know if Jeff, if you want to go on.
Oh, there he is.
Oh,
go ahead, Mark.
Problems because I'm online with the smartphone, and it's not so easy to unmute and such things. And
well, glad to hear your voice. Glad to hear your voice. And I didn't know if you wanted to I just wanted to give you a chance to see if you had any questions that you wanted to ask of anyone here, and Loreen, and just wanted to introduce you both there, to have you both here on the same screen, at least together. Our two different cases of cure.
Hi, Mark.
Nice to see you, Mark. And Gus, a shout out to you, sweetheart. Hello, hello, hello.
Hello to San Francisco.
So Jeff,
when I was last in San Francisco, I hadn't the time to reach out to you. I hope that will be in the future.
Indeed. You're welcome. Send me an email. Jeff, you can get it from Jeff Taylor and me.
Oh,
I have
give you up.
We were in contact.
And by the way, Mark, I'm in Sacramento. I don't live in Sacramento. I mean, in San Francisco.
And
San
I
Francisco.
don't live in Düsseldorf.
Oh, you don't? we have something in common. The San Francisco patient label happened because San Francisco is where I donated the final tissue biopsy donation from the gut. And Dr. Deeks and Dr. Walker shared, I think there were 63 SNPs, and both Boston got half and UCSF got half. And so then thus the name San Francisco patient was born, not the Sacramento patient. Yeah.
I think it's very important that we try to give tissues and support the scientists. And I will fly to New York soon to donate lymphocytes and tissue from my skin tissue. There's the project from Jonah and Lish. They want to search what happened with us. So the project is with Adam, the London patient, Paul, the City of Hope patient, and the Oslo patient also was present in New York, but he wants to stay anonymously. So
Yes.
that
Yes.
he will donate the tissues.
That's terrific. I
Thank you.
thank you both for
Yeah.
all your generosity. It's just astounding, over the years of what you've endured and then what you've done for science and continue to do to support the field. So we'd like to thank you again for joining the call, for doctors you and Peloso for joining as well. We appreciate everybody for hanging in there. I know we've gone about
Yeah.
15 minutes over time, but thank you for sticking it out. This has been recorded and will be posted online. Michael posted the link, and
That's
we'll be
right.
sending that out afterwards to everyone who registered for the call in case they couldn't
That's
make
right.
it as well. So again, thank you so much.
Thank you, Jeff. Thank you, Shu and Michael,
Thank
everyone.
Great seeing everybody.
Take good
Mhm.
care.
Bye.
Bye-bye.
Well,
Yeah, bye-bye.
Mhm.